is a young biotech startup that was founded by two professors from the Nanyang Technological University, Jillian and Chuan Fa. And it is based on work that they have been doing together with another professor from the NTU, Professor James Tang. So this research that they have been conducting over several years led to the accumulation of at least 4 patents that have been granted and more than 20 publications, and hence the company. So the technology in Xingzhan, and hence the research from the professors, is really centered around a family of enzymes called the peptide asparaginoligase, or PAL for short. So these enzymes are found in cyclotide-producing plants, and cyclotides are small cyclized peptides between 28 to 37 amino acids, and they have 3 intramolecular effectors, hemolytic properties. And it also can be recombinantly expressed. So what the professor did was then also conducted similar site-directed mutagenesis on this enzyme, and then generate mutants. Based on the family of peptide asparaginoligase, and we use this then to make antibody drug conjugates and as well nanobody conjugates for cancer therapy. In addition, we also use these enzymes to make protein-protein conjugates, protein antibody or protein RNA conjugates, and as well other custom conjugates including carbohydrates or lipids that are really that it provides a site-specific conjugation as the C terminal or at the N terminal, and as well it allows you to control the amount of drug that you actually add or conjugate to the protein or to the antibody. This makes for a rather homogeneous product, and it's quite different from the typical chemical conjugation that you use when you conjugate a moiety either via the lysine or cysteine on the protein. And as well, after the conjugation, it's a very stable reaction process. So the linker, the conjugation is very stable. And there is also a very minimal footprint on the final product, about only 3 amino acids are finally added, which is the NGL. And because of the fact that we have several members of the same family, we can then actually this enables us then to attach different kinds of on the same protein or same antibody using different conditions and different sequences, recognition sequences. So in comparison to what is available in the market and that is used for conjugation either different biomolecules, Xingzhan offers the various advantages that I just mentioned listed on the left side. And when we compare this with the traditional chemical conjugation, which gives you stochastic random conjugation, and as well compared with chemoenzymatic conjugation, such as using bacterial transglutaminase, and also other enzymes that have been reported, such as bacterial sortase A, and also using non-natural amino acid. Xingzhan offers all these advantages which these other methods only cover in part, or in the case of the chemical conjugation, not at all. So in short, Xingzhan is really a startup that provides integrated end-to-end high-speed precision conjugation. So we carry out targeted antibody discovery, as well protein antibody production and purification, And then we also generate chemical libraries and chemical synthesis of the payloads. And these are then linked together and conjugated via the enzyme, the peptide asparaginase lipases. And the way we work is through partners and to collaborators, is then to out-license either our technology directly to the interested party, or we conduct a co-development to bring the compound, to bring the ADC or PDC into the clinic. Okay. So talking about the linkers and payloads for diagnosis and for targeted cancer therapy. So as I mentioned, we are able to use different payloads to conjugate to a protein or antibody. Sorry, it's a bit small, but stand on this side. So we can conjugate RNA, DNA, or nucleic acids to the protein of interest, fluorescent probes, FIDC that I mentioned earlier, radioligands or radioisotopes or chelators, and as well enzymes. And in addition, you can also conjugate different cytotoxic molecules and immunostimulants, PEG, or through azide chemistry, click handle, you can then add this to your protein of interest. In terms of the linker, and this is really more for the development of radiotherapeutics and antibody drug conjugates, the linker that we are able to add on to the protein of interest could be, very straightforward little linkers that are validated in the clinic, or you can have advanced extended linkers with PEG moieties, including branch chain PEG moieties, or other novel linkers that our collaborators have generated. So Xingzai's technology was validated by Lonza, and I'm sure you know Lonza is the world's largest contract development and manufacturing organization. And they're very highly active in bioconjugation with respect to ADC production and other conjugation forms. So Lonza entered into a collaboration agreement with Xingzai, whereby they offer our technology on their conjugation platform to their customers. And we also engage with their customers directly for specific follow-ups. So for the rest of my talk, I'm just going to focus in on the application of our PALs, or peptide asparaginase lipases, on generating radio conjugates and antibody drug conjugates. So firstly, radio conjugates, they are a well-validated and effective method for cancer therapy and cancer diagnosis. And it really comprises 4 parts. So there is the radioligand, which is bound to via a chelating agent. And then 2, the targeting molecule, which recognizes a target protein, which is usually a cancer-associated protein on the cancer cell surface. And these 2 parts are then linked via a linker, which can be quite standard chemistry, or it can be novel chemistry depending on the researchers who are doing the work. So the targeting molecule that recognizes the cancer antigen can be a nanobody. So this is just the variable domain of a heavy chain, and it can also be the SCFE, or FAP, fragments of antibodies, as you know. It can be an FC fusion, or it can just be a small peptide. And so in respect to this, in terms of small peptides, so I show an example where Novartis recently they marketed, or rather they have a peptide drug radioligand that was approved recently by the FDA, and this drug conjugate is really a radioconjugate deuterium-177 linked to the PSMA, or prostate-specific membrane antigen, and it's for treatment of metastatic castrate-resistant prostate cancer. And this drug alone is projected, because it's very new, it was only approved in 2022, is projected to have US $2 billion in sales in a year. So, as I said, we use PAL to conjugate the radioligands or radioisotopes. And what I show here is that in our hands, the PALs are very efficient and very fast. So after 5 minutes, you can see that there's almost complete conjugation of the radioisotope onto a nanobody. And again, in the table, you can see that the amount of enzyme used is very low. The molar ratio is significantly lower than the nanobody itself, and as well the payload. So this potentially can be a game changer, and in terms of also therapy for using radioisotopes with very short half-life. So this could be very useful and would be having advantages compared to other traditional chemical conjugation or enzyme methods. So we're very excited about this, and we're pursuing hard on this to push forward to develop new molecules that we can take into the clinic. Okay, so the second part of what we focus on is really developing antibody drug conjugates, or ADC for short. And again, this is targeted cancer therapy. So ADC, as you know, then is just the antibody that's conjugated to a cytotoxic drug, whereby the antibody binds to a specific cancer antigen that is expressed on the cancer cell surface. So what happens then is that when the ADC binds to this protein or receptor on the cancer cell surface, the entire complex gets endocytosed, and then it moves from the endosome to the lysosome, where then the ADC is broken down, and then it releases a cytotoxic drug inside the cell, and then this drug can now be effective to kill the cancer cell. So normally the cancer drug, the cytotoxic drug that is used is something that in previous times people thought it was too toxic to be used on its own because it would cause systemic tox. So by conjugating it to the antibody, you now have a very targeted therapy. You bring this drug only into the cancer cells and only to the cancer cells with the specific cancer antigen that is expressed on this specific cancer indication. So also, just to throw out some more numbers. So this is an emerging field and it's very hot currently. In total, there are 11 approved ADCs since the year 2000, with 8 of them approved from 2019 to 2022. So you can see it's really getting very, very exciting and very, I would say, very heavily explored. In fact, in 2022, 249 ADCs entered clinical trials, primarily in the US, but also in China. And the best-selling ADCs in the year 2022 was Casella. Is an anti-HER2 ADC, so basically the antibody binds to HER2 on cancer cells and is used for treatment of breast cancer cells. And in 2021 alone, it made almost $2.2 billion. So this is US dollars. And the second most highest-selling ADC is Ascetis, and this is the antibody binds to CD30, and it's for treatment of Hodgkin's lymphoma. In 1 year alone, it made $1.3 billion. So this is a very exciting field, and one reason why we want to capitalize on it as well. Okay. Yeah, so as an example, just to show you, so we took an antibody that binds to a cancer antigen, and then we labeled it conjugated it using our enzymes, the PALs, either to the C-terminal end of the heavy chain of the antibody, or to the C-terminal end of the light chain. And what we have then is that after about 2 hours reaction, you can see that primarily we have most of the molecules are now dually labeled. There is what we call a DAR2, which is a drug to antibody ratio. So basically every antibody has 2 drug molecules. And the drug that we use in this case is MMAE, which stands for monomethyl auristatin E. So it's a tubulin inhibitor, and so it's highly toxic. So what we did then was that in total, we just took the reaction directly off the reaction, the final product directly off the reaction, and use it to test cells. So when you expose our ADCs to the cancer cells that express this cancer antigen on the cell surface, you can see that the cancer cells are very rapidly killed, and the potency of our ADCs are down to about 80 picomolar. So that's a very good number. In contrast, these same ADCs, they don't inhibit cancer cells, they don't express our antigen of interest. So showing really that there is that targeted specificity with respect to the killing effect. And what we did also was that we compared our ADCs with a commercial ADC that targets the same antigen, but has a much higher drug to antibody ratio of 3.5, so almost twofold more drugs attached to the antibody compared to ours, and is a marketed ADC drug. And you can see that our killing effect is similar to this marketed drug, even though we have half the amount of drug loaded onto our antibodies. So we checked the stability of our ADCs, firstly in human plasma in vitro, and we find that our ADCs, those whereby the drug, the MMAE, is attached to either the AD or to the light chain, is very stable in human plasma. And then when you compare it to the stability in mouse plasma, we found that the ADC, where the drug is conjugated to the light chains, is much more stable, depicted by the red line. But the one with the heavy chain is actually quite unstable in mouse. And this is something we can understand because it's been reported that in mouse serum, a high level of carboxylic peptidase can lead to cleavage of the specific linker that we've used in this ADC linker complex. And that's the reason why with the heavy chain ADC, the linkage is quite exposed because it's at the terminal end of the molecule, and this allows the carboxylic peptidase to cleave at the heavy chain end. Conversely, when you attach the ADC to the light chain at the C-terminal, it seems to be in a privileged position. Let's just go back. When the drug is linked to the C-terminal end of the light chain, it seems to be a privileged position and is protected to a great extent from the carboxylic peptidase present in the mouse serum. Okay, so now comes the interesting part. So we take these ADCs, and then we want to ask the question: do our ADCs prevent tumor growth in mice? So what we did was then we took the same cancer cells that we had done the killing assays with, and then implanted them into mice. And then when the tumor grew to a certain size, we then treated them with our ADCs, those which where the MMAE is conjugated either to the heavy chain or the light chain. And as well, as I mentioned, we also compared it against this commercial ADC that recognizes the same cancer antigen, but has a higher drug to antibody ratio. So these drugs are then dosed as single dose into the mice, and I'm going to show you the data. Yeah. So when the mice were dosed, just one injection, 1 milligram per kg of these 3 different ADCs, we can see that, okay, so the red line is really, the red line or red square represents our ADC where the drug is conjugated on the light chain. The green triangle and green line represents our ADC where the MMAE drug is conjugated on the heavy chain. And purple and is really the marketed ADC where it is conjugated by chemical conjugation. So right off the bat, you can see that at 1 milligram per kg single dose, the green line and red line are much flatter compared to the purple line, showing that at this point, our ADCs are killing the tumor cells much better than the commercial marketed ADC. And this you can see on the other side where you can see the tumor growth which has been excised from the mice at the end of the experiment. So the commercial marketed ADC at 1 mg per kg, you can see the tumor size are quite big across the 9 mice that were treated. And in comparison, our ADCs, the red and the green, really at 1 mg per kg, the tumor size are much smaller than the control. And of course, PBS is a control where the mice were just treated with PBS throughout the experiment. Now, at 3 mg per kg now, at a higher dose, then you can see, in fact, our ADCs led to regression of the tumor. And in fact, that's why when the scientists try to excise the tumors from these mice, they really hardly found any of the tumors, particularly in the mice treated with the light chain ADC. Or those treated with the heavy chain ADC, you can still see some remnants of the tumor. But then, more importantly, in comparison, the marketed ADC at 3 mg per kg, you can see significant tumor growth still. Yeah. So I hope then through this set of experiments I've convinced you that our ADCs are stable, they can perform better because of the fact that the conjugation is very stable and is homogeneous. And then it can lead to tumor regression and better performance even compared to ADCs which are chemically conjugated. So what we're doing now, is really to build up a pipeline in Xinjiang to have our own novel ADCs to novel targets and as well to well-established targets such as HER2, B7H3, and some novel targets which I can't disclose, but then we're using them to treat, the intention is to then use them to treat cervical cancer, colorectal, etc. And as I also mentioned, we're also very involved in developing nanobody conjugates, and we have EGFR radioconjugates and as well additional ones in the pipeline. So finally, we have gathered together quite an impressive scientific advisory board. John Lambert, he's a pioneer in the ADC field. He was the former Chief Scientific Officer of Immunogen, and he was a developer for Casella, as I mentioned earlier, the best-selling ADC in the world. And then Charles Dumonte, he is an oncologist, and he's also very much involved in blood cancer and blood cancer therapies. And Anthony Tocher, he's an oncologist as well, but he's also very much been involved in clinical trials for ADCs. Together they offer a lot of advice in terms of the direction where we should go and how we should proceed. And finally, Nick Devote, he's with the University of Brussels, and he's also someone that we're collaborating with for the nanobody development, radio conjugate nanobody development, and he's working directly with patients and the hospital. So we're excited to work with him and really hope to bring the therapy to patients soon. So with that, I thank you and
thank you, Dr. Lin. Very nice original research, and it will have a good application in the future regarding to target therapy and diagnosis of cancer. Anyone have one or two questions?
Hi. How do you identify the loading of radiopharmaceutical on the antibody, like when you are conjugating antibody with the radiopharmaceutical, maybe before labeling with the radioactive material or after that one? But how do you identify the percentage of load?
Yeah. So I think we do purification. So we do the reaction in the single pot. But after that, we will run it through a column to purify out, based on size exclusion, those which are conjugated, and then we can remove those which are not conjugated, as well as remove the free radioisotope.
Size exclusion?
Yeah. So, yeah, maybe I can also ask Julian to explain that.
Yeah, it's a fair question. I'm not sure so this is done in collaboration with this group on Nick Devote, and they have all the radiochemistry ready, so they do scintillation to count how much radioactivity is put on the nanobody. So it's by scintillation.
So you can, but like with the time, after how much time we have to stop the reaction and go for the treatment or like that?
In 5 minutes. The reaction you do in 5 minutes. The reaction time conjugation time is 5 minutes. So it's like fluorine is like half an hour half-life. So it gives time to still have the radio conjugate ready for going to the patient.
Thank you.
Okay, thank you. Now we come to the next lecture.
Yeah, congratulations for the nice talk, and I could see very significant data that we're discussing too. So really very exciting. I could see one of the conjugates that you have tested in mice even, and the data looks like very interesting. So at what stage that conjugate is now?
So which one? The one in animal studies?
Yeah, animal studies, yeah.
So, yeah, I think what it is now. So right now what we're doing is actually doing in vivo PDX models, because we've done checking the regression of whether it can kill tumor from cancer cell growth in mice. But now we're using patient-derived cultures or organoids, which are then implanted into mice. And then we're going to test and make sure that it actually can kill the patient-derived cancers as well. Because as you know, cancer cell lines are old, right? I mean, they may have complications. So people would like to see also the patient-derived organoids, which are primary cancer growth tumors, and we can further characterize that. In addition, we are also doing more PK studies as well.
Yeah, that's very interesting data.
Yeah.
We will be happy to collaborate with you.
Oh, yes.
Okay, any question can be discussed during the lunch time? Thank you thanks so much. Now we have next speaker, Professor P.K.
I request Chairperson to felicitate Dr. Lim.
So, good morning everyone, and first of all I would like to thank Professor Verma for kind introduction and Professor Rathi for inviting me for the talk here, and I hope I am audible enough for you people. I don't need a mic because our class size is quite bigger than this, so with your due permission I will keep it here. When I will go I will take it with me. So, thank you Professor Rathi for inviting me here, and it's a beautiful place. So, giving a lecture is one part, and another part is looking around the cities also. So you gave an opportunity for us to roam around the beautiful city of our Bharat. So today I am going to talk about reduced selective it will not work. Green only works. You don't need pointer and yeah, so we can just okay. So now one more work is less. So today I'm going to talk about the reduced selective chelpination of oxygenone and oxygen. So what basically we are going to so this is basically chelpigenation through the CH activation, because we know like activating any molecule or particularly some functional group or let's say like CH bond is not that easy. In some cases, not in all cases, some hydrogens are very active and some hydrogens and carbon bonds are very inert as well. So in case of active, we don't have to do anything, it's active, so take any nucleophile and attack this nucleophile on electrophilic center, and very easily replacement would be there, and we will have a desired compound in our hand. But with the inert molecule, right, so inert CH bond, there is a problem, how to deal this. So there is a concept which has been launched in the, let's say like 20 years back, that or even quite earlier also, that you can activate this bond by using some metal, right? So if you provide some metal, then this CH bond would be no more a CH bond, it would be a carbon metal bond which is quite electrophilic, and then nucleophile can attack on it and very easily we can do the chemistry. The same concept So this RH bond, and this RH bond means that CH bond, and we can use this metal, and then C metal bond would be this, so organometal compound would be generated, and then with the functional group, we can replace this metal with the functional group, and we will have a final functionalization. So this is the basic concept which is utilized, and based upon this concept, we have already used this concept for our purpose. So with this, we use this concept and we utilize it for formation of carbon sulfur or carbon selenium bond, right? So with this, we use this concept to use our purpose. So there you can see that this aromatic CH, sp2 and CH bond, which is not at all an active one, which is quite inert. So how to make it active? So use some metal, and then you can have a metal complex, like as it was said there, that carbon metal complex is better for having an electrophilic substitution because it makes more electrophilic center. So with this concept, we can have a carbon metal bond, and then we can use any of this nucleophile to attack on it, and we can have a functionalized product. So with this concept, we use this, and we utilize this concept for our purpose for generating carbon sulfur or carbon selenium bond. So first we will talk about the carbon sulfur, and then we will go to the carbon selenium bond. So carbon sulfur, why we were interested in the carbon sulfur bond and carbon selenium bond? Because like most of the plant and it must be have sulfur containing compound, which is really important for us, which actually solve many purposes. So this is the just broad look that where you can see, I mean, okay, you have to see by yourself, there are sulfur bonds in between, and which is important for us. So why we choose this? Because you can see that it is very important for the medicinal aspect and pharmaceutical aspect as well, because they can be used for the anti-cancer, many more this pharmaceutically active ingredient as well. So these are some molecules by which you can see like sulfur bonds are there, and they are really in a good trial for various diseases. Right. So now come to the point, the challenges that how to deal this. As I said, like you can see once again the problem. So you can see that this bond energy for this CH bond is, even though it is sp2 CH bond, so bond energy 473 joule per mole, it's quite high, and if you compare it with this sp3, it is 410. So even though this it is sp2, but the bond dissociation energy is quite high. So it is not that easy to dissociate this bond or break this bond. So that's why we need to have something which can come in between and break the bond easily. So this is the really problem of this CH bond. So with this, there are other challenges also, that when we do this, this nucleophilic substitution reaction or electrophilic substitution with aromatic compound, there is always a challenge of how to remove this challenge, because there can be ortho substitution, there can be meta substitution, there can be para substitution. So all substitutions are possible in this case, and then inertness is also. So if you are really forcing your system to do the work for you, then in that case the reduced selectivity would be a biggest challenge, because if you force the system to react, then the reduced selectivity would be one challenge. So both the things are there, inertness is there and the selectivity is there. All these three would be more or less equally have an equal this bond dissociation energy. So how to deal this? That's why we took some functional group which can assist, as it was shown there, that it can assist by forming this Pleda cycle or some metallo cycle, so that this bond can be very easily dissociated. So these are the problems which we have to encounter. So for all this concept problem, and we use this concept for our purpose. So we took this quinoxylene molecule, and And there you can see the nitrogen in between. So the nitrogen will help in forming this metal carbon bond in between, and then very easily it would be more electrophilic. And so there would be two concepts which would be, so one would be we would be utilizing it for the sulfur, and another would be for selenium, right? So both the thing would be going in the same direction, so with the same reagent as well. So it's really hard to read, I think. So anyway, so quinoxaline and quinoxanone. So you can see this molecule that they are quite active. So for the different purposes. So we chose this molecule to derivatize further so that we can utilize it for our purpose. So there are certain activity of this, and we can use this concept and we can utilize it for the purpose. So what we did is that we started explaining how to activate this bond, and then do this replacement or nucleophilic substitution on it. So first of all, in basic chemistry, what we do, we first screen the solvent. We did the screening of the solvent, and we found this dioxide water, 1:1 equimolar ratio of this was working best, and 57% was obtained with this silylated product with us. So we obtained, like water as a better solvent, because you see water, with the water 94% of the yield was obtained, and the rest of the time it was not as good as compared to the water one. So with this we could very easily say, like, yes, we can do this reaction in a greener way, let's say, like, or less toxic, greener. I don't know what I should use or not, but yeah, less toxic environment is there. So with the less toxic environment, water cheaper, at least we can say, less expensive. So with this solvent we moved further for the reaction, catalyzing other, optimizing some other catalysts as well. So we use different catalysts with the palladium, with the rhodium, with the ruthenium, all the catalysts, even nickel and copper. So all the different kind of catalysts we utilize for the purpose, right? And with this screening, we found that the rhodium catalyst is best with the CP dichloride. So with this, we were obtaining the 94% in water. So we no other solvent, as you can see, no other solvent was suitable for the purpose, right? So with this, water as a solvent, rhodium as a catalyst, we move further and screen the additive. So you can see that we move changes the different amount and concentration of the additive and the oxidant as well. So both the thing we change at the same time, right? And we found that the additive silver as a 3.3 equivalent is the best in presence of oxidant silver, and we were obtaining it's in a better rate, that is 94% yield. So with 0.9 equivalent of this silver and the silver carbonate, we obtained the best yield. So we were really lucky in the first go that whatever the amount we took, it was the best, right? So we got the best yield with the 94% yield, and with this we found that yes, changing a different loading of the additive or the oxidant, we found that this is the best that we can use 0.3 equivalent, 0.9 equivalent of silver and as an additive and the oxidant, and we could obtain the yielding at 94%. So with this, we wanted to see that whether this method is applicable to different substrate or not. So we did substrate scope analysis and screening of the compound. We did changed all the diselenides, and with different selenide you can see that it was giving even 95-96% of it. So it was really giving the excellent in almost all the cases where we try to change the substrate scope with this diselenide with different electron withdrawing group, electron releasing group, and the effect of that. So in all the cases with the electron withdrawing, electron donating group, it was giving the better rate, and the compound was nicely characterized by all this spectroscopic technique as well as the crystal analysis. So there was no problem in getting the structure here. So with this, then we also wanted to see that whether we can vary the quinoxanone or not. So with this, we vary the substrate score with the quinoxanone, and in all the tried cases, we obtained the better yield. Right. So with this, we established that yes, this protocol is going to work for silylations. With all the diselenide, and all the compounds were obtained in a good yield. Right. Then we wanted to see that whether this can be transferred to the sulfur or not. So with the same diselenide, we use this with the sulfide, and with this disulfide also, it was giving better yield in all the cases. So you can see, it was, I mean, we know like this is a little bit not active as the selenium. So that's why the percentage of yield is not as good as it was in case of selenium. So, but anyway, the same protocol was working well with this disulfide, and the disulfide we could obtain the different compounds with the different sulfides in a nicer yield. Not only that, you can see here the use library was generated, and every time it was giving a better yield, and even all the quinoxanone was utilized, different quinoxanone was utilized. So either the disulfide or the quinoxanone, both the side, with the sulfur or selenium, we were obtaining the compound in a better yield. So with this, we wanted to see that whether the synthesized compound can be used further or not. So we did, like all the latest stage functionalization. So we did, because in all the cases there are several drugs which is in a market. You can see on the left hand side that they are the drugs, and we wanted to see that can be very easily synthesized with this protocol. There is no problem in getting this product. Right. So we didn't optimize in all the cases that what is just we wanted to see that this protocol can be extended to the known pharmaceutically active ingredient or even the drug. So it was delivering the compound in a nicer rate. So it was tried with all the drugs which is known, and in all the cases it was giving the product in a nicer rate. So potential of this protocol was also evaluated in our case, and it was working well with this. So with this, we wanted to see that what is the mechanism of the reaction, and for that we performed a certain control experiment, whether it is following the ionic mechanism, whether it is following the radical mechanism, what is the mechanism of the reaction. So we performed several control experiments by excluding one another reagent to see that what is actually role of each and everything, what is the role of the catalyst, what is the role of the oxidant, what is the role of the additive, what is the role of the solvent. So all the things were screened nicely, and with this control experiment we could establish the mechanism of this reaction, that how it is working. So as usual, it is working in a simple concept. With the first, you have to activate your CH. So it is activating CH by forming this metal complex. So first thing is our metal complex. So oxidative addition took place. So first thing is the oxidative addition, and then the transmetallation, like selenium compound could go to the cycle. So second step is the transmetallation, and then third is the elimination, and then obtaining the product. Because my pointer is not working, it is not visible, so it's like what to explain and where to explain. So I will do fast. So oxidative addition for making it making it better electrified, and then the transmetallation, reductive elimination, and product was obtained in this way. So this is how we have studied, because there was two possible ways by which two possible paths was also possible, and we are not sure that which path is possible, whether this path B is working or path, because both look similar. We could not isolate the intermediate because it was not possible to isolate the intermediate, and so this reaction works in this way. So with this, I can summarize it like, yes, we can very easily use this protocol for same protocol for introducing sulfur in the molecule, selenium in the molecule, and we can obtain the products nicely. So this is the first part of the talk, and now come to the second one. How we can introduce this sulfur and selenium into the benzoxazine. So with the same concept, whatever we used in our case, so we use it for our purpose. So we know like the significance of the molecule of benzoxazine, what are the properties of it, and so on. So with this, this is your significance of this molecule, why we chose this molecule to work on it. So this is how we chose, because this benzoxazine is also well-known pharmaceutically active molecule, and with this very that we kept in mind that yes, this is molecule which can be used further, because you know the Manila was looking for the molecule and doing the chemistry. So I mean biochemistry, in fact. So you know, we have to synthesize compounds so that the biochemist can take it further. So with this, we took the same concept, and we with a different simple benzoxazine molecule. And here you can see there is a nitrogen which is going to assist this nitrogen, because we need some functional group, or let's say some atom which can somehow assist in the chemistry. Right, here you can see the nitrogen is there, and that can assist the chemistry, because it will, you know, like, because choosing a molecule for the reaction, you first have to see what is my molecule, how it can assist my chemistry. So there is a nitrogen which is, because if you recall the last mechanism where there you could see like this nitrogen would be assisting to form a metal complex, right? So this should be the thing. So because this is actually leading to this bond formation, right? So with this in mind, we could use this concept, right? And we're using this nitrogen. You can see here the nitrogen. So with the nitrogen, we could utilize this nitrogen for our purpose, because it will form a cyclometal complex, and then this CH bond would be activated and then can work further. So with this, we use the same concept which we used in our previous cases, and we screen for the solvent temperature in this case, and we found that the DMF is the right. So with DMF. Right, so with DMF at 120 degrees centigrade, we could operate end in 89%, right? And then we are better.
Next slide. Next slide.
So, चलिए कोई बात नहीं, आप बढ़ाते रहिए घटाते रहिएगा, ठीक है। कहाँ चलो पीछे जैसा। So with the DMF at वही रहो भैया, बात करोगे तो काम कैसे चलेगा? या काम बोलोगे या तुम बोलोगे? ठीक है। So with DMF at 120 degree, we could obtain the product in 89% because increasing the temperature, right? So when you increase the temperature, we could obtain this, decomposed product, right? So decomposed product was there. We observed that way. you can see that at 140 degree centigrade in the same DMF, we could obtain the yield in only 64%. So sometimes the chemistry which we have studied, like increasing temperature by 10 degree.
रेड का बैलेंस ज्यादा उसमें नहीं काम करेगा।
हाँ ठीक है चलिए शुरू कराते हैं जितना करेंगे उतना हम करेंगे ठीक है सो अच्छा हाँ so what I was saying that here sometimes increasing temperature is not always rewarding, and here was the same thing that it was not rewarding. So it was the product was getting decomposed, and that's why it was giving the lesser yield in that case, right? Next, screening of the oxidant and the catalyst. So we try to use different catalysts in this case as well, and we could not obtain good yield with this benzoxazine with rhodium or any other ruthenium catalyst. It was working only with the palladium, not with the other this transition method. So with the palladium, we wanted to see that which palladium salt is good for this transformation. So we could obtain this palladium acetate as a better catalyst for this, and then for oxidant, copper bromide as best combination. So palladium acetate, copper bromide, and which was giving the compound in the better rate. So it was giving the 89% with the palladium and copper bromide. Right. Now we move further for the screening of the amount, the concentration of the catalyst, right? So we could vary the concentration of the oxidant and the catalyst as well, and we found that 10 more percent of the palladium acetate and 1.5 equivalent of the copper bromide with the 24-hour we could obtain the So two questions, right?
Yes.
One is the role of silver oxidative. So you can see oxidative addition, right? So oxidative addition, you need to oxidize the bond. So there are additives which can be, like they can assist in forming this, they are working for the purpose. And the second is that with this dye, like H2O2, let's say, like peroxy acid or something. So no, we didn't, but certainly it is not going to work with them because they will generate the radicals, right? If they will generate the radical, we don't know whether how it will work. So if they will generate the radical, it may directly attach over there, right? So O bond can be formed in that case. So O something. So that kind of chemistry is also known. Like you can use this peroxy acid or something, which can be easily transferred.
But in place of this dithio or dicellulose,
Yes, yes.
it is very smelling compound. In place of we can in this case thioester can be used.
Then you will generate a different compound.
This in the place of SPS.
Yes.
In place of SME or SPS, SI group transfer from thioesters also we can, you can try this maybe.
Yes, you can try, but you are not sure that you are going to get, because with this disulfide, right, you have a huge variety of substrate reagent, you have a more accessibility of those reagents are higher over there. But with that, you have a certain restriction, but that is a good solution. You can try, of course you can try.
Because this is very smelling compound. We can try this one and go also it won't slightly form. Thank you, sir.
Thank you, sir.
Yes, Dr. Nice. You talk in simplified manner. And the questions we will restrict because we are having short of time. So thank you.
Thank you, sir. I would like to chairperson facilitate Dr. Sen.
Students are requested to be seated, please. Be seated, please.
Now we have done next. We have Professor from again Delhi University. He did his PhD from Good Institution Scientific Research Institute, and he pursued postdoctoral research at various parts of the world, including Germany, Europe, and he has more than 100 research papers and authored many books and chapters. His research focuses on carbohydrate heterocyclic synthesis, and recently he is a research professor at University of Japan. I invite Professor Ram.
Good morning everyone, and I would like to thank Professor Verma for inviting me so nicely, and just like for inviting me to start here. Sir, you think I need to take this? If there is no need, I would like to continue with the other. So it's required. Okay. Yes. Thank you for inviting this nice place, and we have already visited one place yesterday. So today I am going to deliver the talk, and we will continue this process after this. So actually I'm going to talk about some chemistry where I'm going to use this acetophenol derivatives to synthesizing new class of heterocycles. So that was the work which is going in my lab, and using this as a beginner, we have synthesized, I don't know, almost all the heterocycles, all the carbocycles we have generated just starting with this acetophenol. So we have used this molecule to synthesize either some precursor, or we have directly used this compound as a reagent, and we have generated a new class of molecule. So nothing to say about this, because this molecule is so important. I think even if you start with graduation, you start with this molecule and you start learning chemistry from this. Either you start the condensation reaction, you start reduction, you start some oxidation reaction. So there are many types of reactions which have been done just by using this molecule, because this carbonyl group, this is itself very useful, because as soon as it is present, it can act as an electrophilic center. You can involve different types of the nucleophile which can attack over this, and it can give you the new class of molecule. This can be as a part of aldehyde, ketone, ester, amide. So there are different variety of the molecule where this carbonyl group is going to be present. Then this molecule is already used for the synthesis of large number of precursors, heterocycles, carbocycles, and conjugated compound. And the most important thing which we have focused, I think this is working, but this red color is not specified on this here. So the ketone, which has the alpha hydrogen bearing carbon, those are going to be the acidic in nature, and by using the different type of the nucleophile tools, you can just generate the carbon ion that can be used to generate the different type of the chemistry. So using this as a beginning of the molecule, we have synthesized different class of heterocycle. I have just discussed two class of molecules here. And out of that, the first one which I am going to discuss is the 1,2,3,5 tetrahydroimidazole 1,2,8 pyridines. And here, before going to start about its chemistry, I would like to discuss a basic property of this molecule. You know, this is the combination of two heterocycles. One is imidazole and one is pyridine. So when pyridine and imidazole fuse together, having one nitrogen common in both, then you are going to generate this class of heterocycle. And Dr. B.K. Singh has already discussed, and I think all of you are aware that when you are going to look at any drug, I can say that more than 90% drugs are heterocyclic. And out of that, the nitrogen containing compound, they are having the best pharmaceutical properties, and they can, you can see that there are many drugs, and they do have the nitrogen, like the multiple number of nitrogen or one number of nitrogen, having different type of the heterocyclic ring. So definitely this molecule also have the different type of the pharmaceutical properties, and they are also used because they do have very good quantitative properties. Because of this quantitative property, they can also be used to generate different types of the organometallic compounds, as well as because of this property, they are also used in the medicinal chemistry, because you can use their coordination property, electron donating, electron withdrawing capacity of this molecule. So I have just shown some of the molecules over here. These molecules, many of them are already done, and some of the molecules are already in the clinical trial, and they are used for different type of the biological activity already. So now I'm going to come on the synthesis, but before going to start our procedure, this is some of the procedure where we have to incorporate the imidazole with the pyridine. Basically, the main processes which are available in the literature, the 2-aminopyridine is the primary precursor for this, and using this 2-aminopyridine as a primary precursor, they can use different type of the reaction approach, where they can insert 2 carbon and these 2 nitrogen to fuse the 5-membered ring to generate this imidazole pyridine ring. And for this, they can use a multi-component reaction, they can use that condensation cyclization reaction, they can use some other approach. But in our case, what we have done is, we have started this chemistry. So the beginning is estophenol, or you can say the aryl alkyl ketone, and then we have generated this ketene diethyl this ketene diethyl, which is going to act as 1,6 acceptor. So you have mainly heard 1,4 Michael acceptor that is mainly used. But what we are doing is we are using 1,6 Michael acceptor as well as 1,4 Michael acceptor. So in our case, there is always possibility the competition between 1,4, 1,6 as well as 1,2 Michael addition. 1,2 addition. So out of these, depending on the reaction condition, nucleophile, there is a different mode of the reactivity. So here we have synthesized this molecule that is 1,6 acceptor, and then we have used the cysteine. Using the cysteine, and in the simple reaction condition, like you just have to use the DMSO, potassium carbonate as a base, you have to mix it, stir at the room temperature, and you can end up to this multifunctional compound. So as I said, we have to start from this acetophenol, or you can say the aryl alkyl ketone, and this is the first step where we have to generate this alkene nitride as a starting material. And this is simply well known in the literature. You can just use the ammonium acetate in acetic acid and toluene. You have to reflux. You have to use Dean star because usually this reaction is running in a large scale, so you have to keep removing the water. If you don't do this, then there is a reversibility of the reaction, and you will enter to the less amount of the product. So by this, we have synthesized all this alkene nitride. Then we synthesize the ketene diethyl of this compound. And this reaction is also done just by using the carbon disulfide and methyl iodide in sodium iodide solution, and at low temperature 0 to 5 degrees centigrade, and we can generate this ketene diethyl. Now you can see that considering the cyanide, so cyanide is going to be 1,2 acceptor, then the alkene nitride, that is going to be 1,4. Now the center where there is the sulfur is attached, 2 sulfur, they are going to act as a 1,6 acceptor. So this is the optimization of the reaction condition, and I must admit that what we were thinking, we haven't got that product. Our thinking was something else. When I will discuss the mechanism, then you will see that what was our expectation, and what further happened. Because the left nucleophile, that is the S-methyl anion, is already in the solution. And because of that S-methyl anion generated in situ in the solution, we have entered to a different product, which we will see in the mechanism. So here we have just screened a simple reaction condition because sulfur, SH, that is also very contained acidic proton, so we don't need to go for very strong base. But yeah, we have to have some cyclization in the other nucleophile also, which will generate in situ. So we have to use the base, and we have used like triethylamine. From here we have started, and you can see that in THF it didn't work, but as soon as we go for the polar solvent like DMF or DMSO, at room temperature we gone for the 20 and 50% of the yield. That means the mild base is working, but definitely we have to go for further optimization. So when we went for the potassium carbonate, in that case we have got 75% of the yield, and then using, because polar solvent was working well, so we used this polar solvent and we taken the cesium carbonate, and we gone for further strong base like KOH, and we found that if you go for further strong base, the reaction is giving some problem. That means product formation might be observed, but this compound, they also have the acidic proton adjacent to the carbon. So that might give the side reaction, and after the formation of the product, it might undergo decomposition. So at last, this potassium carbonate and DMSO we consider the best, and we just use this reaction condition and we synthesize different functionalize this individual pyridine compound with a different functionality. So as I said, this data matches with the data what we were assuming. But when we got the crystal structure, then we found that no, this is not the one what we were thinking, this compound is something else. And now if you see the mechanism, you will realize that why we were thinking something else. So here what was happening is the cysteine, it is going to add by 1,6 Michael addition to the carbon where the 2 sulfur is attached with the formation of intermediate A. And this intermediate A, when the reversal of the negative charge will occur, then there is possibility of the removal of all these 3 S-containing groups. So major possibility with the S-methyl goes out, and then you get the intermediate B, and then cysteine is attached over this. Now this nitrogen, which is present in the cysteine, that nucleophilically again attack, because this is an intramolecular reaction, so that is more facile. This again attack over this carbon by 1,6 Michael acceptor, and then we get this intermediate. So what we were expecting, that after the formation of this nitrogen will cyclize with the cyanide, and it will give you the thiazolopyridine. That was our expectation. But since in our reaction the S-methyl is already present, and what that S-methyl did, that S-methyl attacked over this carbon, which is just next to the sulfur, and there is a ring opening. So this thiazole ring open, followed by the cyclization of this imine, which has been generated by attack of this nitrogen to this ester group. So what happened? The thiazole ring has been opened, and this imine again cyclized with the carbonyl group via consecutive movement of the electrons, and we have got the intermediate. When this O minus falls back, then ether, you can say the OET minus goes out, and we got this compound 5 as a final product. So actually some of these molecules has already given us very good activity, and that activity is still in the process, so I haven't gone for that activity, but yes, we have gone for this molecular docking study. And then this methyl group containing this indisciplining, they are giving 2 different activities. Here we have shown on the DNA gyrase, and on DNA gyrase, this compound, and there is one more compound that is thiophenol substituted, that is giving us activity even better than ciprofloxacin. And we are having the anti-cancer activity in nanomolar concentration that is already under process, so we have not considered here because still study is pending. We cannot just disclose over here, but yes, so docking study is also comparable, and we have got that is compensate that result what we are doing. So this is the next part, and in this part we have generated the quinoline and isoquinoline molecule, just again starting from this istophenol, and generating some molecule which is going to act as a precursor, and here the multiple steps have also been carried out in a single step. So that I will also show. Okay, so okay. So again, quinoline and isoquinoline. I think you might be knowing the drug which is based on the quinoline as well as isoquinoline. So the pharmaceutical property of these molecules. So these are some of the examples where this quinoline and isoquinoline molecule is present in the pharmaceutically active compound, and they are also part of the natural product as well as the pharmaceutical ingredient. And these compounds, they have very much fluorescent property depending on the incorporation of the different type of the group. So we also have used this isoquinoline and quinoline as a precursor, because whatever molecule we are synthesizing, they are able to incorporate heterocyclic group also. So in that case, if we are incorporating the heterocyclic group involving this quinoline nitrogen as well as isoquinoline nitrogen, we are able to coordinate with the metal, and that is making this molecule as a molecular sensor. So that has also been done with the collaboration of some of our friends. And then they are also part of the agrochemicals, corrosion inhibitor, chiral ligand, and also there are many asymmetric ligands which are involving this quinoline and isoquinoline as well. Their part. So now, in order to synthesize this molecule, we have to synthesize another 1,6 microreceptor, that is 2'-pyral with a different functionality. The functionality why it is required, because if you don't have that specific functionality in your molecule, then your molecule may not be reactive, because you have to increase the electrophilicity of some center by putting some electron withdrawing group, and you have to control the electrophilicity of some center by putting some nucleophilic group like amine. So here we have synthesized this starting material just by using this methyl cyanide acetate and carbon disulfide and dimethyl sulfate in presence of this base, and we are able to synthesize this ketene diethyester, which reacts with either cyclic ketone like tetralone, 1-tetralone, 2-tetralone, and different acetophenol. This can react under the basic condition to give this 2'-pyral or coumarin molecule with the S-methyl functionality. And this S-methyl functionality, because S-methyl is very good leaving group, if this is attached with a double bond which is conjugated with the electron withdrawing group. So in this case, if you are just adding some secondary amine and you just mix in the ethanol and reflux fermentation 3 hours, you can just replace this S-methyl and you are able to incorporate this secondary amine over this position. So since this S-methyl is very good leaving group, we usually try to reduce the electrophilicity of that center, because you have multiple centers for the reaction in this molecule. So now by putting this nucleophilic group, at least we are reducing some possibility of the side reaction. So now just by using this precursor, you have used this pyridine 4-ol, and the protection is required because this amine that is also nucleophile. So we don't want to have the competition between nitrogen nucleophile and the carbon nucleophile which can generate by the carbon adjacent to the carbon molecule. So in this case now, we are able to synthesize this isoquinoline, the Bob protected isoquinoline, and in this case the sodamide in DMS2 was the best, and we have got 89% of the yield in this case. So just by using this reaction condition, we are able to synthesize the simple aryl functionalized quinoline, as well as if you are going to have the 2'-pyral from the tetralone, you can have the polycyclic compound directly from this compound. So you can see that in the last we have synthesized the polycyclic compound also, which has the nitrogen in the array. So now in the next step, you can just go for the reproduction of this compound, which is quite known procedure. You can have any acidic component and you can just stir it, and then you can just filter your compound without any problem. So we have just used acidic condition to deprotect this, to have this tetrahydro isoquinoline as a product. Now after having this tetrahydro isoquinoline, we thought, okay, let's go for the conversion of this tetrahydro isoquinoline to quinoline molecule, and for that we have to go for the oxidation. And the simplest oxidizing agent we know is the DDQ. So we thought, okay, let's reflux with the DDQ in toluene and we may get the product. But surprisingly, when we are using this DDQ in this condition, it was not working. And not only DDQ, then we gone for some other oxidizing agent, because this NBS and AIDN we have also optimized for some other reaction. that was also not working. Then these condition, and then there are other oxidizing agents have also been tried, but somehow that was not giving us conversion to this quinoline. So we have to go for this, and then we have found out some new procedure where we can just convert this compound, the bark protector. That means we don't have to do the protection. We can just start with the bark protector. If you want tetrahydroquinone, you can just deprotect the bark at lower temperature. If you directly end up to the isoquinoline, you can just end up to the isoquinoline just by using iodine as a oxidant in DMSO. So I think you know that DMSO in iodine is very good oxidant, and that has been well known and well explored. Even you can find many reviews on this. So just by using this combination of DMSO and iodine, we have just optimized this reaction condition at different temperature, and we have found that if you are going to use 10 mole% of iodine in DMSO at 140 degrees centigrade, we can just enter directly to the isoquinoline. And if you go for lower temperature, then we are getting some of the quinoline, but there is leftover deprotected compound. So that also suggests in the mechanism that first there is a deprotection, and after that there is a oxidation to give this compound. So after getting this condition, we are able to get this compound. So we thought, since we are synthesizing our quinoline molecule in DMSO, so why not just start with this pyran and directly end up to the isoquinoline. So what we did is, we directly taken this two compound, and first step we carried out, and once we observed the compound by the TLC, that now it's in the solution, we added iodine to this. And we just raised the temperature for this, and surprisingly, now we started from this, and we directly end up to the isoquinoline in one part. So you can see that here we have done the ring transformation, then we have gone for the deprotection, and we have gone for the oxidation. So here the multiple step has been carried out in the same part. And just by using this, we have synthesized these compound, and this is the single crystal structure for this compound, which has also confirmed the structure. Now, since we are able to synthesize isoquinoline, so we thought let's focus on the quinoline also. Maybe this procedure works for quinoline, and yes, this procedure works for the quinoline, and surprisingly, this reaction is much more faster. It was not taking as much as time in case of the isoquinoline. So this reaction was faster, and we have also synthesized different quinoline molecules just starting this 12 pyran 2 moles. So again, there is transformation, deprotection, and oxidation was carried out in the same part. This compound was also confirmed by single crystal X-ray. And this molecule, so you know, the quinoline molecule is also known for the anti-cancer activity. So we have also gone for the molecular doctoral study for them. And in this case, the two compounds, one is the naphthalene one and one is the thiophenol containing quinoline, they have shown a good result, and they are also in the process of the in vitro activity of the compound. So this is the some of the interaction which has been shown over here. So now this is the third one, and again this is another heterocycle which has been generated, that is the pyrazole. So pyrazole, which is very easy to synthesize and very useful molecule. They are also versatile, known for the medicinal chemistry, drug. They are also known for the material chemistry because they do have the very good property. So here again what we did is we synthesized this pyrazole with the dicarbonyl functionality. So you know, the dicarbonyl compound is also very good precursor to generate different types of the heterocycles. Like Professor B.K. Singh has shown that he has synthesized some compound where we are trying to do the CH activation. So he used the diamino compound Now you can use the dicarbonyl compound and you can generate those compounds. So this is some of the introduction about this, that they are known for antimicrobial, different type of activity you can see, anti-cancer, anti-proliferative, anti-hyperglycemic, as well as kinase inhibitors. And they are also acting using the metal organic frameworks and ultra-porous materials and luminescent material also. And the most interesting that now this is the first compound, because we are looking in the literature and we haven't seen any pyrazole with the dicarbonyl containing compound. So this is the first compound where the pyrazole with the dicarbonyl. So you know, like you can just have the phenyl benzyl ketone. You can just do the oxidize and you can synthesize the benzene, which you use for your chemistry in the lab, like benzyl benzylic acid rearrangement and different type of the chemistry. So this is the first case where we have incorporated adjacent to the pyrazole. And then we are looking at the literature that how usually you have to modify the pyrazole. So you know the pyrazole has very few positions and very few possibilities to modify it, because there is two nitrogen, one is completely hybrid, and one has, like you can go for some electrophilic reaction, and that leftover position you have to incorporate an electrophile and nucleophile. Then you do have to use the metal catalyzed approach as well as the different half-reaction conditions to do that. So these are some of the reactions where they are trying to either synthesize pyrazole and or they are using the pyrazole as a precursor, and then they are trying to incorporate the different type of the group in this molecule. So in order to synthesize this molecule, what we have to do is we have to synthesize this ketene diethyl, and then again we treat this ketene diethyl with this acetophenol, and we have generated again a new molecule where there is another acetyl group is left over with this 1,4-Michael acceptor, and this molecule was synthesized in quite good yield. There was no issue. And after synthesis of this, what we did is we have just performed this reaction in two parts. First, we have just taken the DMSO hydrogen and we just cyclized this molecule, because we can see in the mechanism also that how it is happening. So it can add by replacing this as methyl, and it can condense with the carbonyl. That is one possibility. Or this hydrogen can condense with the carbonyl first, then intramolecularly it can add at the carbon where the S-methyl is attached by a Michael addition, and then S-methyl will go out to give you the product. Just by using this, here we have isolated the 4, and 4 was converted to 5 in iodine in DMSO, because this method was already reported in the literature to synthesize the benzene. So since as we have already seen that we have to perform this reaction in DMSO, so we thought, okay, what we will do is we just synthesize this compound in one pot. We will generate the pyrazole, and in the same pot we will convert this compound to the dicarbonyl compound. So considering that fact, we have just used this hydrogen and different type of the oxidizing agent which can just convert this CH2 to carbonyl. And we found that when we are using this hydrogen first, after conversion of this pyrazole, we add iodine at 80 degrees centigrade. So we are using to get 72% of this dicarbonyl containing pyrazole, and this was the best reaction condition over here. So doing this in one pot, just by starting with this compound 3, we synthesize all these molecules in moderate to good yield. And now this was the synthesis with the simple hydrogen. Now we have thought, okay, let's take the functional hydrogen like phenyl hydrogen or methyl hydrogen, because when you take these hydrogen, then there is two possibilities. They can be isomeric, because there is possibility that methyl can go at next position where this dicarbonyl group is attached, or there was alternative possibility that we can get the methyl which is at position 3 with respect to the dicarbonyl group. but surprisingly and interestingly, we have got very selective compound, and this compound 6 we have got from this methyl hydrogen, and then this compound 8 we have got from the phenyl hydrogen. So the compound was selective, and it's not only with the simple hydrogen, we can go with this compound for the functionalized hydrogen also. And this is the confirmation of structure by single crystal X-ray. So just to check whether these carbonyl are as good as the benzene. So we have also done this reaction using a phenylalanine diamine, and we are able to synthesize phenylalanine functionalized pyrazole. And now this compound is also in the biological study as well as the material study. So now we are generating the di-coordinating ligand like nitrogen of pyrazole and nitrogen of this phenylalanine can coordinate with a different type of the metal, and it can give you a different type of the complex which can be used for a different type of the chemistry. So now this is the mechanism, and mechanistically what we propose that first there is condensation of this hydrogen with the carbonyl group, and after condensation the next nitrogen can attack by a Michael addition to the carbon bearing s-methyl with the formation of this pyrazole by loss of s-methyl group, and then after formation of this pyrazole, this can tautomerize to compound 2. Now this position which is adjacent to pyrazole can be iodinated. This is also known. There is nothing new. If you have the iodine can iodinate that position, and you might use the iodoform reaction also. So you can iodinate this procedure to get this iodo compound. Now the matter comes how it goes to the dicarbonyl. So this can interact with the dimethyl sulfoxide. Oxygen of the dimethyl sulfoxide can replace this iodide ion and form this intermediate C where this oxygen is going to be a positive charge. Now the iodide ion can attack over the sulfur, and it can imply this oxygen, and at last there will be loss of HI and dimethyl sulfide with the formation of this dicarbonyl compound, and we will end up to the this pyrazole bearing dicarbonyl compound. So with this, now I would like to conclude everything in one shot. So I will include here only 3 molecules where we are trying to do like 1 pot, 2 pot, multi-component reaction in same pot. But this is only just few. If you are going to look at the literature, and even if you see the paper from our group, you can see just by using this molecule, you can just name the heterocyclic compound we have already synthesized just by starting with this histophenol. You can say pyridine, pyrimidine, pyrazole, oxazole, thiazole, you just name it. That is synthesized from this paper. So that was the beauty of this compound. So yeah, now we have shown that we have developed the transition metal free. Why is it transition metal free? Because when you are going for the transition metal, you have many limitations. Limitation means we should have to be the rich. If your lab is not rich, I don't think it's easy to perform the catalysis. Then you should have more facility. You can see that here, if you have that rotavapor, magnetic stirrer, and some round bottom flask, and these reagents you can go with it. But for catalysis, you have to have the nitrogen atmosphere, then maybe the glove box is required, then there are many other special conditions are required for that. So there are things, yeah, okay, so now we have generated this, using this 1,6 Michael acceptor, we have synthesized this imidazopyridine, as well as we have synthesized this iodine and dimersopyrid functionalized multifunctional pyridine, isopyridine, as well as pyrazole in one pot. And these molecules, they are still under investigation for the biological activities, so we have to learn from that. But with the document result, we have got the promising result for this compound. So that was overall for today. And now these are our collaborators. So Dr. Gaurav, he is doing antibiotic compound, and Professor Rajendra Mitra is doing biochemistry for us. Dr. Thanendra Yadav, he is doing molecular modules for us. Professor Han and Professor Ismail, he is doing the biological work for us. And then these are our students who were involved in this designing this molecule and synthesizing this. And now, the most important, now our University of Delhi via IOE, they are giving us the fund that is helping us to do this research. So I must thank University of Delhi, the CSIR, ICMR has also funded for this work. And at last, I would like to thank University of Delhi and Student Facility Center about Delhi University to helping in this development of this new chemistry. So thanks all of you.
You don't have any questions because they are
Thank you.
I request Professor Verma to felicitate Professor Amit Prakash.
प्लीज इस तरह से हल्ला ना करी, कोई लेक्चर दे रहा है।
So the topic of my presentation is catalyst control, CS functionalization and emulation of heteroarenes. In here, before starting my presentation, one thing I want to clear, like Professor V.K. Singh sir has talked about the CH activation, but I am talking about the CS functionalization. Both terms have the similarity. Both terms have the similarity, but there is the difference between the CH activation and CH functionalization is that the directing group and the catalyst. In case of CH activation, we must have to use the directing group and the catalyst. Like here, so many master students is there also might be. So maybe you study about the protecting group. So when we protect some of the functional group in the in any molecule, that one is going to change in the term of the directing group, and that directing group activated the neighboring carbon wherever the directing group is present. But in case of the CS functionalization, directing group may be required and may not be required. Both is possible. If the reaction is happening in the presence of directing group and with the transition metal catalyst, then the term is known as the CH activation. And if without transition metal catalyst, if we are using the CH functionalization, the chemistry is known as CH functionalization. So both cases is the CH functionalization, either CH activation, either without CH activation. So what is the CH functionalization? CH functionalization means the replacement of the either breaking of the carbon hydrogen bond by the heteroatoms such as nitrogen, sulfur, oxygen, phosphorus, halogen. Like B.K. Singh sir also present the sulfonylation, means chalcogenation. But here I would like to present the my chemistry using the pi unsaturated compounds like the olefination, alkylation and alkylation, either allylation. So what is the CH functionalization? In traditional approach, whatever we use in the lab, when we have to functionalize any particular functional group at the particular position, that time it requires the various steps. But in case of CH activation, if we have to functionalize the molecule, it requires only one step, either two steps. We can get our desired product. Like in the case of this first example, here there is no directing group. That's why we have to make the acid oxidation. That time we are getting the mixture of the compound, ortho, meta and para. But if we have to functionalize at the particularly at the ortho position, that time when we make the directing group, then we are using the transition metal catalyst, we directly get the selective product at the ortho position. That is the major product, means the regioselective product in case of the CH activation. So during my research, in my research laboratory, I am also performing the CH activation. Whatever work here I will present, all is my work, none other work. So in my research laboratory, I have also tried the CH activation and CH functionalization both strategy to get the biologically active compound. Because for any chemistry, if you are doing any simple synthesis, that is not the, I think not sufficient. You must have to focus on the drug molecule, so that we can applicable that molecule can be go for application in the industry, medicinal industry. So like this one, disubstituted indole, naphthoquinone, and this one also emanated naphthoquinone drug molecules. This kind of molecule I have synthesized in my research laboratory. So the application of the CH activation is in industry and in the natural product synthesis also. So by using the nitrogen as a directing group, we can use several molecules, several atoms as a directing group like oxygen, sulfur, nitrogen. These kind of atoms we can use as a directing group. So in my chemistry, basically I have focused on the nitrogen as a directing group, and by using that nitrogen as a directing group, I have utilized so many pi unsaturated compounds like various olefins, diazo compounds and alkynes also. And by using that chemistry, I have synthesized so many molecules like 8-methylquinoline, indole, oxyindole, isoindolinone, phthalamides, etc. So how many types of CH activation is possible? There is 3 kinds of CH activation is possible. One is this is addition type reaction, another one is the SN type reaction, and third one is the dehydrogenate type cross coupling reaction. So in my research laboratory, I have tried all kinds of reaction. So very few examples I would like to present here. One I will present the recent one, and one is very old one. So here, in addition type reaction, I have utilized the carbonyl compounds, olefins and the alkynes. So this is the first example for the addition type reaction. In this reaction, I have utilized the secondary benzamide and aldehyde as a coupling partner by using the rhodium catalyst. In all my chemistry, whatever I will not present the optimization reaction condition for the transition metal, either solvent or any other things. I will directly go with the molecules. So when I use the secondary benzamide with aldehyde, I get the 4-hydroxy isoquinolinone compound. And then we think about what only if I will synthesize this hydroxy isoindoline compound, that is the meaning of nothing. So I think about the synthesis of the drug molecule. So on going through the literature, I found this chlorothalidinone compound. This compound is reported around 10 steps by using the traditional approach. But through my chemistry, when I utilize this N-benzylated amide, And when I use this chlorosulfonyl aldehyde compound using rhodium catalyst, I have directly this chlorothalidinone compound. This is anti-hypertensive drug and this is the marketed drug. Next, about the addition type reaction, I have utilized the andazolone compound by using the melamide. Melamide is itself biologically active compound and used in various drug candidates. So when I use this one, I make the several drug candidates like Spiro type compound using the rhodium catalyst. So in this case, in my research laboratory, we have synthesized the salicylic oxide compound. That is also the drug candidate. This is non-steroidal anti-inflammatory drug molecule. We have synthesized the estrogen. We have also synthesized this metronidazole compound. We have synthesized this fluorescent for using the pyrene compound. This compound used in the treatment of the anti-cancer molecule. This is used as an anti-cancer drug molecule. So this is the reaction mechanism for the synthesis of the drug candidates of the Spiro compounds. Firstly, the rhodium metal makes the active metal catalyst which undergo for the CH activation process, make the 5-membered metallocycle intermediate, and then undergoes for the coordination with the melamide, and then make this in species B. And this species B, in the presence of acetic acid, provided the alkylated compound 3AA upside, and this 3AA compound undergo for the even CV type elimination and provided the olefinated compounds, and this olefinated compound undergoes for the isometric type reaction and provided the spirocyclic compound. So to confirm the reaction mechanism and actual formation of the intermediate, we have also isolated this complex, rhodocycle complex, during the reaction. And after the isolation of this one, to confirm this one, we have also take the XRD analysis of this one. So this confirms that during my reaction, this intermediate is formed, and after the formation of this one, whole the reaction is carried out. Next is the SN type reaction. So for this one, we have tried we have utilized the allyl methyl carbonate as a coupling partner and pi unsaturated compound. So for this one, we have used the alpha beta unsaturated carboxamide and another aromatics compound. Only one or two are listed here. So by using this one, here we have tried the ruthenium as a catalyst, and by using this one, we have made the allylated compound. So after getting only the allylated compound, that was not sufficient. That how to get the any drug molecule for this after the synthesis of this one. So we have what we did. We make here aminated at the para-amide compound we have utilized, and then when we have used the ruthenium catalyst, we get only allylated compound. So we have further screened the metal catalyst, and during the screening of the metal catalyst, we found that rhodium metal coordinate perfectly with the nitrogen and activate the neighboring carbon and make the indole derivatives with the allylation. But with ruthenium catalyst, we get only allylated carboxamide compound. So this was the difference between the ruthenium and rhodium catalyst. Next, for the addition type SN type reaction, we have also synthesized the xanthone and chromone aminated compound. As we can see, xanthone and chromone are very important drug molecules utilized in the cancer treatment. So we have also focused on the aminated synthesis of the chromone and xanthone derivative, like in the presence of the ruthenium catalyst. So we have made around 50 examples for the chromone and xanthone aminated compound by using the different donating and withdrawing groups and several drug molecules. So after the synthesis of our desired compounds, we have evaluated the anti-cancer activity in compared to the doxorubicin, and we get very nice biological activity, anti-cancer activity of these compounds in different cancer cell lines. This is our very latest chemistry for the SN type reaction. Here we have targeted for the synthesis of amino acid, because amino acid are very important biologically active molecules and directly used in the drug and in the synthesis of proteins and several natural products. So for this one, when we have directly used the amino acid for the reaction of the amination, we failed. Around 100 reactions we have tried, all is failed. Then I think one day why not if I utilize the azalactone, and azalactone, if we will try for the ring open, that can directly make the amino acid. So we have tried the, one time I put the reaction without my using the student. I directly make the azalactone, and when I put the reaction, I get this aminated compound with azalactone. Then I use the various catalysts to ring open, failed. So suddenly during the literature survey, I find out if I can use the urea as a catalyst, asymmetric urea. When I tried, I directly get the aminated amino acid compound. That is very nice compound, and by using this amino acid, I have synthesized around 20 drug molecules which is under the animal treatment in the industry. I have collaboration with them. So now, like we have made the aspirin, astrone, benzodiazepine, this one, I cannot read from here. And there is so many drug molecules which we have synthesized. Might be maybe possible, you can see from there. These are the, and then we have confirmed the formation of this molecule through the XRD analysis. And this is the reaction mechanism. Like first the metal, make the active metal catalyst. How to reaction perform? First important point is the activation of the metal catalyst. So whatever we are using the additive, either oxidant, first they make the active metal catalyst. Like if we are using the rhodium with silver additive, silver give the electron, plays as a ligand role, give the electron to the metal and make the active transition metal catalyst, which undergo for the CH activation process, and after the CH activation make the 5-membered metallocycline intermediate, which undergoes the coordination with that metal species and make this imine intermediate B, and then after the protonation reaction we directly get our desired product. To confirm this reaction also we have isolated the formed complex during the reaction, and this complex we have confirmed by the X-ray. There is the sum of example to get the for the ring opening of the azalactone and to get the amino acid by using the urea catalyst. Like here we have made the this valine and alanine derivatives. Now next is the dehydrogenated type cross coupling reaction through the CH activation. That one we have tried for the radical type reaction. So for this dehydrogenated type cross coupling reaction, we have tried so many coupling partners. We have used the aldehyde, we have used the alcohol, ester, alpha keto acid, diketone, carboxylic acid, benzylamine and toluene type compound. So for this one, firstly we have tried the N-benzyl triflamide. We make the N-benzyl triflamide and we activate this neighboring carbon hydrogen bond, and then we for this one we have used the aldehyde compound, and by using this one we get the acylated N-benzyl triflamide compound. So not only N-benzyl triflamide, after making this one we think how to make the nice chemistry for this one, so that we can make the applicable for the industry, either for the medicinal purpose also. So we have through the literature survey we have found this neopan molecule. This is an analgesic compound. This is the marketed drug, and this one is reported around 20 to 30 still. But when we have tried our chemistry, we firstly make the methylation of this N-benzyl triflamide and we make the reduction, and after the reaction we use the acids, and after the intramolecular cyclization reaction we directly get the neopan compound. So we have also tried the dehydrogenated cross coupling reaction by using the alpha keto acid and through this chemistry we have made the 3-isochromine compound. We have also made the acylated oximes, and through this compound we have also shown the application in various drug candidates. Next chemistry is the azobenzene. I have focused in my research group. I have also focused on the azobenzene chemistry because azobenzene is very important molecule and directly can be used in the CH activation because there is the 2 nitrogen to activate the particular carbon bonds, CH bonds. So for this one, and this is very nice fluorescent compound. Very nice fluorescent compound and used in the treatment of the cancer disease. Directly used in the treatment of the cancer disease. So, but the challenge was only azobenzene, only one molecule. So first we think about how to make the different kind of azobenzene to show the derivatives. So for this one we have synthesized around 20 azobenzene compound, and for this reaction important point is the control the selectivity. Here you can see the inversion, very fast inversion in there, cis and trans. So important point is how to control the cis and trans position. So that's why we have selected the particular, first we optimize the transition metal catalyst to control the selectivity. So when we use the ruthenium catalyst, we get both cis and trans. But when we go with the rhodium catalyst, we get the only trans selective product to get our desired product. So here, from the azobenzene, we can make several drug molecules also. Like here you can see the 2H-indazolol containing compounds, indole compound and sinolene compounds. All are the drug candidates. So based on this one, we have synthesized we have tried so many chemistry with this one. Like here, we have published around 10 research papers using the azobenzene, and we make many heterocyclic drug molecules. Like here, with azobenzene we have tried the alpha keto acid, and we make the 2H and the acylated 2H-indazole compound. We utilize the melamide using the azobenzene. We get the indole compounds. We use the sulfonium. We have tried the vinylene carbonate. We have tried the dihexazolone also to get the aminated compound. We get the alkene to get the indole derivative. We have tried the aldehyde to get the acylated 2H-indazole compounds. So after the publishing all this chemistry, I have write the review which is published in the tetrahedron in 2018. So I would like to select, I selected only one, last one, to show the mechanism of this chemistry. So here, this is very interesting chemistry. Why? Because when we use the electron donating group with the azobenzene, we get only 2H-indazole compound. But when we have used the electron withdrawing group, that time we get the 4+2 type cycloaddition reaction and get the sinolene compounds. So to find out how this one is happening, we go in the deep to study about the reaction mechanism. Here I don't present. We have also did the DFT calculation to find out the exact mechanism of this chemistry, and we have also isolated the two of the intermediate from here, which I didn't present here. These two intermediate. One is this alkylated, and from here this intermediate. This one. But we get this product. So from this one it confirms that the sinolene is also going to form. So here this is the reaction mechanism for this one. One is the 4+1 type cycloaddition reaction and another one is the 4+2 type cycloaddition reaction. So first the rhodium undergoes to make the active metal catalyst in the presence of the silver salt and the oxidant, which undergoes for the making the metallocycle intermediate after the CH activation, and then cyclic carbonate go for the this olefin insertion, and then after the migratory insertion we get this alkylated compound. And this alkylated compound, if we are using the electron donating group, that time it goes to the 4+1 type annulation reaction and make the 2H-indazole here, attack on this tertiary carbon, and then ring open with the release of the CO2 and directly get the 2H indazole, hydroxyalkylated 2H indazole compound. And when we use the electron withdrawing group like NO2 or S star, that time we get this phenolene compound. That time this we observe this lone pair attack on this carbon and making the phenolene compound. So this another chemistry from my research group. This is very interesting chemistry and my favorite chemistry. I have published 20 research papers only on this chemistry. That is the C7 functionalization of indolic compounds. That is the indole derivative. Functionalization of indole at the C2 and C3 position is very easy because of the pK value of the C2 and C3 is very lower in compared to the C7 position. That's why how to solve and how to get the C7 functionalized indole compounds, we think about if we can functionalize the indoline at the C7 position and we undergo for the oxidation process, that time we will directly get the C7 functionalized indole compounds. So for that one we have, and the challenge was what? The pK value. The pK value at the C7 position is around 43, that is very high, and then C2 position is 38. That's why the formation of the metallocyclic complex at the C2 position is very easy and the 5-membered formation is very feasible in compared to the formation of the 6-membered metallocyclic intermediate at the C6 position. So through this challenge we have tried so many reactions and we get this acylation, alkylated and tandem cyclization also. This tricyclic compound we get directly by using the directing group of carbamyl. We have made this olefinated compound and this aminated compound. So through this one we have also synthesized around 10 drug molecules, like which one is presented here. So this is the combine of this chemistry. Like we have tried the, and all of the chemistry published in very high impact paper, high impact journals. So we have tried the olefination reaction. Like we have, when we use this hydroxy olefinated compound, we get the alkylation. We use the aldehyde, we get the acylated compound. We use the alpha keto acid, trifluoromethyl olefin compound, and then ester. This paper also published as a cover page and very VIP paper through the editor choice. And then we have also reported the allylation of the indoline at the C7 position. And after making the C7 functionalized indoline, we directly go for the oxidation process and get the C7 functionalized indole derivatives. So this is some of the recent chemistry from our research lab which we are doing already performed. Published this paper. Like we use the this azomethionamine, first one. Azomethionamine by using the vinylene carbonate, methylene carbonate compound, and through the first this reaction make the hydroxy-allylated compound, and after the formation of this hydroxy-allylated compound undergoes for the cyclization reaction and make this tetracyclic Bridges compound. To confirm this one, first we think this is just a simple tetracyclic compound, but when we go through the characterization, NMR, that time almost the same, so we confuse. But when we check the XRD, and we write the paper also for the tetracyclic compound, but when we get the XRD, that time totally changed. This was the Bridges compound. Then we again write the manuscript and we publish this one in Organic Letter in 2022. This is one of also very recent strategy. No one till now, no one has utilized this this methylene thiazolidinone compound, and this is glitazone. This thiazolidine compound is known as the glitazone. Glitazone is very important, the anti-diabetic molecule, and used in the type 2 disease, type 2 diabetic disease. So we have directly utilized this one as a coupling partner, as a olefin source, and we make various drugs. Around 50 drug molecules we have synthesized using this one. We have combine the two drug molecule, one is the glitazone and one is other drug molecules like salic oxy and so many drugs. So two is the representative example is here. This work first we have published, tried in the SCS catalysis, but due to the lack of the evidence of the mechanism, that paper was rejected and they suggested something. We have tried the DFT calculation and we made the this paper in the organic level. And we have also used the MBH adduct as a olefinic source, and by using the MBH adduct, we have made this indeno pyrazolone derivative, first one. That paper we and with this azomethyl imine. This chemistry also come from the that chemistry idea come from this one, that we can make the cyclic compound same like upper one. This right side first one and left side first one. Both idea is almost same, but the chemistry is totally different. That the formation of the product is also different. So here first one, this one, when we use the MBH adduct, we make this pyrazolone compounds. That paper we have published in Angugam in 2019. We have used the MBH with benzylamine. All that benzylamine is the marketed available, commercially available. We make the benzodiazepines, annihilated benzodiazepines. We have published this work in ACS catalysis. And then with this anoxide, we have published this Bridges type compounds. Now recently, and by using this first chemistry, we have made this estrone and this steric oxide compounds also. That is the very important drug molecules. So recently we have now focusing on the transition metal free catalyst. Because there is so many drawbacks and the expensive chemistry, so we move to the transition metal free catalyst by using the ylides. Ylides are till now reported to make the either tricyclic compound, either for the making of the olefins. So what we did, we used the ylides to make the direct alkylated compound, neither the cyclization, neither the olefins. We tried the olefin ylides to make the alkylated compound. So from this chemistry, we have this quinoxaline, make the methylated quinoxaline, either alkylated quinoxalines, and by using this chemistry, we have synthesized these natural products only in one step, and this compound is reported in 15 steps previously. But through my chemistry, I have reported the synthesis of this compound. This is very important natural product only in 2 steps. One time alkylation and then again alkylation. That's it. This is another chemistry which we have made this anoxide for the alkylation chemistry by using the this ylide chemistry, and we have synthesized so many alkylated drug molecules like hydroquinone and phosphodil also. These are the my research paper which published as a cover page. Around 60% of my research paper is published as a cover page. So just very few I want to show here. And I would like to acknowledge the Department of Chemistry, Delhi University, because I did my PhD. My professors are seated here. It's my pleasure to present my work in front of them. Professor V.K. Singh sir, R.P. Singh sir, Shailendra sir, and so on. And secondly, I would like to acknowledge my postdoc supervisor from 2013 to 2015. I did my postdoc with Professor Insoo Kim. He is very nice guy. I learned so many chemistry, so many things, how to live like the human beings also, how to behave with the research scholars, how to behave with the students. So many things I learned from Professor Insoo Kim. He is very nice guy, and he helps to me. He helps to me to help the Indian student to go for the postdoc. Around 18 students, 18 बच्चों को इन्होंने मेरे कहने पर help किया जो postdoc के लिए गए. Some of in US, some in South Korea also. So he is very nice guy. I really appreciate him, and I would like to my current department, Lucknow University, Department of Chemistry, Lucknow University. I also would like to thank my funding agency, NRF South Korea, and now currently the sir who provided me the 2 research grants around 60 lakhs, ServSoil and ServSRG. Thanks to all of you. And currently I have one PhD position available. That last date is already over, and interview I will going on 16. If somebody is interested, can apply. Thanks to all of you.
Innovative and for really translation medicine. I don't think
I will be happy if any question.
No question, please. Thank you.
I request the chairperson to felicitate Dr. Neeraj.
last speaker, Dr. Leonardo Oliveira, distinguished scholar with a bachelor's in chemistry from Federal University of California. He is presently a research officer at Federal University, and he is research across his politics, synthesis, heteroscience, carbohydrates. He has published more than 200 research papers and had more than 1,000 scientists. Dr. Leonardo, you have 20 minutes. Please finish in 20 minutes. Thank you.
I will try.
my strategy is work with single molecules. What's happened?
फुल मोड करते हैं
Sorry. So with this collaboration, I felt okay. So I work with this best glycerol sugar, triazoles, lactobacillus, and some drugs, and try to find collaboration in biological areas. With this strategy, I reach a bunch of results. So it's not rational drug design. It's serendipity, it's true, but this worked well because I need to find results, experimental results. Talking molecular talk is good, revision some results is good, but finally I need to do work in the laboratory, experimental results, vitro, in vivo. So this is my research project that I focus today. You can see sample molecules, sugar, naphthalene, nucleic bases, and glycerol carbonate. That we can produce this from glycerol. The first step is glycosylation reaction. Open the carbonate cyclically to arrive finally at naphthalene derivatives with sugar. On the other hand, we can change the nucleic bases, like thymine, and incorporate some sugar triazole to obtain more complex molecules. So this is a background with naphthalene. Naphthalene is sometimes not very good because naphthalene is toxic. But we synthesized this molecule the first time in our laboratory. We obtained, of course, some results against parasites and also against some cytotoxins, against some cancer activity. This is a strategy with naphthalene that I prepared these derivatives, our research groups. You can see 3-4 steps to prepare this compound. Here also 3 steps to obtain this compound. But we don't have results against malaria with these compounds. But on sample strategy of infection in functional groups, we obtain this compound is similar but not the same. And we found good results in vitro and in vivo malaria diseases. So this let me think about naphthalene. We can sometimes use naphthalene. We motivated to explore also some compounds against Zika virus, like these compounds. We tested all these molecules with naphthalene, sugar, buoyant, triazole. All these compounds show less potency against Zika virus than these compounds. So what's changing here is we have a portion of sugar, heterocycle, in this case is triazole, and phytalimide group. This problem is Zika virus in my country, my city, Recife, in Pernambuco, northwest of Brazil, is very, very hard for us, because the main problem, in my opinion, is these children with microcephaly. Until today, you have this problem. So, motivate my I can say my best collaboration with Professor Lindomar, that work in Philippines. This is new strategy at this moment in my laboratory is working with glycosyl bonds or glycosyl reaction, glycosylation reaction, and click with. I'm trying to find a new method, but easy methods, because if you produce some molecule, the biological molecule, you need also find pathway is to produce this molecule. If you have 10, 15 steps, it's not good. So I try to find 4 or 5 steps maximum in my preparation. So this is recent results that I have prepared from the glucose and glycerol enable research to obtain this kind of compounds. The best results that motivate my work with carbohydrate chemistry restarted this subject is the reaction when we produce this compound, this glycosyl compound, from glucan and glycosylated glucan and glycerol carbonate. You have a minimum 2 possibilities to obtain 4 products: alpha beta, selectivity, and the R or S R or S configuration in this position. Because here is a mixture of R and S configuration. So we have a chance, because I reached this compound, this poor diastereomer, by resolution, by crystallization. It's like pasta. It's very, very difficult to reach this. So I think so this is a chance. I had a chance with this procedure. The compound crystallized ethyl acetate some 2-3 hours. It's very good. So this is configuration ionics. So now I have all this strategy to prepare a new compound based this new scaffold, carbohydrate and azido glycerol. So then you can expand more complex molecule from this other preparation. You have here oxide as well. So you can see these more complex molecules. So these results, recent results from these molecules, we try to find some molecules against tuberculosis. And during this process, we found also some results in anti-cancer activity against lung cancer. It's a surprise for us. So you need to think the intermediate compounds, sometimes you have good results. So don't forget the intermediate compounds. So we increase, in this case, the complex structure to produce all the structure to obtain the more bigger molecule. And now we test for good resistance stem from tuberculosis. The next step for the work. can see here the diplexer, the structure of diplexer with nuclear bases. You can see here. And the result is less than 10 micromolar against the Zika virus. So you have some 2 or 3 linear compounds to filter study. These are some presentation about the carbohydrate-based drugs that launched in this period. Is not can change this, and the carbohydrate-based is shown down very important scale for just antiviral to use it as an antiviral compound. So I will show very fascinating these results. Okay. So these results now, I have this question because I have this compound that is good results. You can see an octagonal structure in this compound. So more time, this compound is toxic because octagonal is toxic. But you have good results against tyrosinates, these compounds, and I think now this compound have not viral activity. I don't not yet find some relationship with these results and the viral activity. I don't know if this exists. But the synthesis is fast. More time we have the glycerol derivative and heterocycle compounds like benzo, tiro compounds and naphthanones. This is a second strategy to clarify other compounds. This time without the glycerol parts. It's spaced by glycerol and no spaced by glycerol. You find these good results for these molecules. Against melanoma cancer also. Okay. Against melanoma cancer and also anti-tyrosine. So you compare with logic acid and dogs. You see that the result is very, very good. So antiviral results with these molecules is also very good. So you have this compound, like a leader compound. You can see sugar, moiet, glycerol spacer, triazole, and the heterocycle, benzoheterocycle compound. So maybe you have a dual activity. Anti-cancer and antiviral for this leader compound. You can try to change some structures with this molecule. So the second result is to prepare this kind of compound. We try to prepare first the triazole and after that the glycoside, but the yield is very low. And the selectivity, alpha, beta selectivity is very low. So we change the sequence of the reaction. We start for first the glycosylation and after the triazole compounds. So we have the best reaction alpha, beta, and best yields from all the compounds. The problem still is the toxins from naphthol molecules. So we change the naphthol that you know is toxic to phytolamide that we know is not toxic. So for these results, you have good results against FG2 cells and the cytotox against neuroblastoma cells and virus cells. So all these molecules, phytalimide, triazole, and the carbohydrate saturated, you have good results in screening against Zika. So this is, you have 4 compounds less than these 10 micromolar. So this is good for to try to find a new leader compounds for the next step. In vivo testing, for instance, you have 4 products with this structure. And in general, now we have these good results, and we know that you test, pre-treatment test, that the virus, you can know if the virus will enter in the cells to replication. So our molecule will not stop this, but the results show that our compounds avoid the replication inside the cells. So you avoid this replication and the results. So the next steps to finish, we use the same molecule against SARS-CoV-2, and we have also good results. This is the best molecule. And to conclude, we have, you can see a bunch of molecules that have bioactivity. And that's Zika virus, SARS-CoV-2 in this case, this left case, and against Zika. I will still try a test for SARS-CoV-2. Okay, so I want to acknowledge my grants for BRICS project. This project permits me and my stay here. So it's very good. CAPS and FASEC is fellowship for the students. And this is the student that work with me, collaborated from Professor Arnaud from France, University of Orleans, and my best friend, Celso, the big man, is chemist in Rural University. Lindomar is there. This is Hati and me. So this is my group. And the major is master and the doctorate students, PhD students. So this is my dream in Brazil. Strand Orient. You can see that. And for us, we like to go to visit. Maybe this beach is beautiful. The south of Pernambuco. And do you know this guy? He's Rati to 2019. This beach. So maybe the next conference will be Rati. Okay. We'll try.
Thank you, Professor Ronaldo. Nice talk. Any questions? Can we ask?
In final molecule, if carbonyl group converted to sulfur, then it can be good binding with Zika virus. Can be found.
Change the molecules?
Yes, final molecule, this carbonyl group converted to carbonyl, C double group convert to thiocarbonyl. Maybe it will more effective than this final compound, whatever it is. So maybe it will go to activity against Zika virus.
Also.
Because carbonyl have thiocarbonyl have good binding energy with Zika virus.
You have a carbon mixer. You think about this, it's just take the azidyl alcohol and change to carbamate. It's easy to prepare it with the phenolphosphine and reaction. So we think about this before, but don't have time to do this. But this is a good suggestion. It's possible to take the carbamate derivatives.
Yeah, because carbohydrate chemistry is very interesting chemistry. But
Yes,
so
it's
much
hard
challenge is
also.
here during synthesis, workup, and so much challenge here. But it's very good chemistry. Yeah, thank you.
I like it.
Thank you. If I possible, I will also try this bio content.
Okay, we can work with it.
In future, yeah.
What about it?
Thank you.
Please give a hand to Ronaldo.
I request the chairperson to facilitate Professor Ronaldo.
Yes. Now look here.
Thank you very much. Okay.
Thank you to chairperson for chairing this session and thank you to all.
Huh?